We're continuing on this lecture on the cell and parts of the cell, and specifically parts of prokaryotic cells. And we just finished up with what's on the outside in terms of the cell membrane and the cell wall, and what you're supposed to know there in terms of the peptidoglycan and the gram-positive, making those specific bacteria sensitive to penicillin and penicillin-like drugs. And then, of course, the gram-negatives, their cell membranes have the O antigen, which is highly chemically reactive in terms of activating the immune system, and is called an endotoxin. Now we're ready to talk about what's inside. And there's not a lot inside a prokaryote as compared to a eukaryote. The first thing you need to know, and this is also important in terms of antibiotics, is that they have a 70S, a ribosome. 70S refers to its size, and I'll give you another number later in terms of compared to what, whether this is big or little. Ribosomes are a combination of RNA and protein, and you don't need to know that. You do need to know that they are responsible for protein biosynthesis. You may already know that proteins are important in your diet, but as far as cells go, proteins perform all the important functions inside a cell. If you've already had biology 201, which is anatomy and physiology 1, you know about actin and myosin. Those are the two proteins that are responsible for muscle contraction. If you've already had biology 202, you know about receptor theory and how, for example, when a neurotransmitter is secreted, it binds to a receptor, and this activates a response in the target tissue, or the same thing happens when a hormone. So receptors are also made out of proteins, things that chemical messengers bind to. So proteins perform all the important functions inside, not just our cells, but bacterial cells. And the ribosome has the duty of making the protein. So proteins perform all the important functions in a cell. And they work not just inside the cell, but some are exported. For example, mucus is manufactured by ribosomes in the cells that line your airway, and it performs an important function of providing some protection to the epithelial cells in your airways and in other places. So proteins are very, very important inside, and in some cases there are some that are also important outside. Now, the way you make proteins is you have to have an instruction for assembly, because proteins are made out of single amino acids. There are 20 different amino acids. All right, so how do you decide the order of assembly in terms of whether you add one specific amino acid, and then which one of the 20 do you pick to put in after that? Well, the order of assembly of proteins, that is dictated by your genetic code. So in terms of making proteins, this starts with DNA, either in your nucleus or just running around free in the prokaryote that we'll talk about here in a minute. So this is your genetic code. The first thing you have to do in terms of us is to get the genetic code out of the nucleus, and so and bacteria use the same thing even though they don't have a nucleus, so you have to read the genetic code and turn it into a slightly different code of what we call RNA. So the DNA, the genetic code, gets undergoes transcription. And then it's turned into a more portable code that then can be read by the ribosomes called RNA. All right, so RNA has several different functions. One of the functions of RNA is as messenger RNA, and this is the one that takes your genetic code to the ribosomes, and then the ribosomes read the code and then assemble the protein based on a very specific order of amino acids as dictated by your genetic code. Sometimes genetic code is off by a little bit, and so then you have a variation of a particular protein that's made, and of course that's how we have some genetic diseases like cystic fibrosis and sickle cell anemia. But anyway, so getting back to this, we start out with DNA, which is genetic code. We have this process that we'll just kind of refer to in a black box way, called transcription. You make a more portable form of your genetic code. So this is like taking your instructions to a Xerox machine and making lots of little portable instructions as compared to having a big strand containing all of the instructions. All right, so or a textbook containing the instructions. So you start out like with a textbook. You take a page that you want to copy, make 100 copies. That's like going from DNA to RNA. Now the RNA, the more portable form of genetic code, goes to the ribosome, which is the topic of our conversation here. The ribosomes then read the genetic code, and they're responsible for translating the genetic code into a protein. Let's do it this way. I want you to have a picture that at the end that makes sense. So the DNA goes by way of transcription to RNA, this portable form of genetic code. The RNA then goes to the ribosomes, where it's read and translated. Into a particular amino acid sequence that then makes up your proteins. All right, and the idea here is there's one gene for every one protein. A slight oversimplification, but it'll work as far as where you're going. All right, and how you go from one gene to one protein is by way of this scheme, where you start out with the gene and the DNA. It undergoes transcription, turns into this little portable copy that can move about more easily. The portable copy, the RNA, then is read by the ribosome and translated then into these extremely important and somewhat easily damaged entities called proteins, which perform all the important functions inside the cell. All right, now this 70S is important because it turns out that eukaryotes mostly have a larger 80S, S standing for Svedberg, size ribosomes. So ours are bigger. So eukaryotes have an ADS ribosome, or have ADS ribosomes mostly, and I'll tell you where the exception is. The ADS ribosome is not only bigger, but there are very specific things that bacteria have developed in targeting other bacteria and trying to kill them, called antibiotics. And the 70S ribosome is sensitive to a number of very important, clinically relevant antibiotics. And of course, you already know how penicillin works, so this is not, we're not talking about penicillin here, we're talking about other, what we call more broad spectrum antibiotics. So these antibiotics work against all bacteria because all bacteria have a 70S ribosome. And examples of drugs that antibiotics are used to kill bacteria based on inhibition of protein synthesis and binding to and blocking the 70S ribosome include chloramphenicol, tetracycline, didn't look right. Rest of it's right. And what I referred to as the mycin drugs, so mycin being a suffix coming from a certain type of bacteria that looks fungal-like. But any drug that ends with mycin, like erythromycin, they would also have this same function of blocking the 70S ribosome and therefore blocking protein synthesis. And therefore, if you can't replace the proteins as they're damaged, the function of the cell is going to be severely compromised over time, and it will die. All right, so like I said, there's some important things that are going on in terms of differences in structure between prokaryotes and eukaryotes that we have leveraged into important ways of treating patients. Other things you find inside a prokaryote, in addition to these ribosomes or free ribosomes, or nucleoid, now remember that prokaryotes lack the karyo, which is a nucleus, so this is a circular strand of DNA. This is analogous to a chromosome, which is a linear form of DNA in eukaryotes. All All right, so this is where all the genes are that we just got done talking about that are transcribed and then translated into proteins. Are found on the DNA here. All right, so this is the genetic code. There's also some smaller bits of genetic code called plasmids. So these are small pieces of extra chromosomal DNA. These small fragments contain generally a few genes, and they may be passed from organism to organism. And at this point, just to reinforce something I mentioned earlier about prokaryotic structure that you find on the outside, this is where you should be recalling these structures called pili, which are modified fimbriae. All right, so remember we mentioned earlier that the pili are used to exchange genetic material, and that would be in the form of these plasmids. Now, why are these important? Well, we just got done talking about antibiotics, and hopefully you know that bacteria are able to unfortunately develop a resistance to antibiotics. They can have or acquire genes that make proteins that, for example, break down penicillin. And if you happen upon a plasmid, if you're given a plasmid that contains a gene for penicillinase, the name of this enzyme that breaks down penicillin, all of a sudden you've acquired antibiotic resistance to that drug. And other types of antibiotic resistances are also passed from bacteria to bacteria, sometimes between different even types of bacteria by way of plasmids. So these transfer genes that confer antibiotic resistance, unfortunately, all right, so this is a bad thing, so we have a sad face down here. But then we can also use plasmids. We can construct artificial plasmids and genetically engineer bacteria to make things that we like. Like, for example, you can go and take the genetic code for the 52 amino acids that make human insulin. You can take the human genetic code for insulin. You can genetically engineer it into a plasmid, put it into a microorganism, and it will assemble in the proper order all of the amino acids to make human insulin. And this has been done, and it's a very good thing to be able to genetically engineer bacteria to make an abundance of proteins that would otherwise not be easily obtained. And of course, the reason why they can read, why a very, very primitive structure or organism like a bacteria can read the genetic code of a human is because, well, we're related. We have the same code in terms of how we make proteins, which is a very, very basic aspect to all living organisms. But anyway, so getting back to this, so it's also important in genetic engineering. And this, of course, with regard to at least the manufacture of human insulin or Humulin. That gets a smiley face, and of course, the bacteria don't charge anything, and then the people in New York, they need a profit, so they end up charging $300 a vial, and then we're back to an unhappy face. All right, the next important structure that you should know about is not found in all bacteria. It's only found in certain gram-positive rods or bacilli, and they're called endospores. And this is the last thing that you need to know with regard to what's inside a prokaryote. So endospores. Endo meaning within, spores being small germ-like elements that can give rise to a new individuals. So these are dehydrated dormant structures. They are important because they can survive incredibly harsh conditions, so you can practically boil these things. No, you can boil these things in certain instances and they will still live. So they can survive harsh conditions. In terms of chemicals, extreme temperature, or exposure to radiation. And they're not really alive, but they will live. So that if you put them then in the right conditions, a year or two later, they will grow. And so they survive harsh conditions like chemicals, extreme temperature, or exposure to radiation, or they can go for a long period of time without food or water. Okay, and then under the right conditions, then they will sort of like come back to life and form bacteria that look like a bacteria and then are able to reproduce. And because they are so robust and difficult to kill, they require specialized techniques like pressure cookers or autoclaves, where you don't just have hot water, but you have hot water in a closed container. And the problem with water is that once it gets to boiling, which is 212 degrees Fahrenheit or 100 degrees Celsius, that's it. It goes off. And so the temperature of the water can never be higher than a certain amount because of atmospheric pressure. All right. So once you get to the point where the vapor pressure of the water exceeds the atmospheric pressure, then you have boiling, and it doesn't ever get hotter than that. So what do you do to make water hotter? You put it in a closed container where you allow the pressure to build up. This raises the boiling point, raises the temperature, and all of a sudden you can get up to 250 or even 375 degrees in terms of being superheated steam. So anyway, so that's what you do to kill these buggers. You have to kill them with specialized techniques like autoclaves, which work like pressure cookers. So pressure cookers and autoclaves have a similar mechanism of action. They're just for slightly different purposes, and of course pressure cookers are generally smaller than autoclaves. All right, and as I mentioned previously, the endospores are found in certain gram-positive cocci, or it's not cocci, but bacilli. And there's two main genera that are important here, the Clostridium and the Bacillus. So you have the one Bacillus, which is Bacillus anthracis. So I thought CIS. Should have gone with my first inclination there. All right, so Bacillus anthracis causes anthrax, which is a very horrible bacterial disease that people used to get because they lived in close contact with herding animals that routinely carry this bacteria. And this is one of the things that Louis Pasteur worked with was the anthrax. And then as far as Clostridium go, Clostridium tetani, which you can tell by the name causes tetanus. Or you might call it lockjaw. Clostridium botulinum. All right, now notice I didn't write out the whole Clostridium. I just abbreviated it C, botulinum. This is an appropriate way of signifying the binomial nomenclature that we talked about before. So it's either genus and species, genus being capitalized, species being lowercase, or you can, in this case, you already know what the genus is, so we're just referring to it as the abbreviation for the genus, a letter C, and then botulinum, which is botulism. A flaccid type of paralysis because you can't release any acetylcholine from your motor neurons that innervate your skeletal muscles, so the nerves can't communicate to the muscles, and so you have a weakness and then eventually paralysis, and eventually you can't breathe. Clostridium tetani has the opposite effect. It causes the motor neurons to release an excess amount of acetylcholine, so you're constantly contracted, hence the term lockjaw. Clostridium perfringens, which causes gas gangrene. Let me again underline this. So gas gangrene is what happens if you have an injury where the tissue becomes necrotic and anoxic, where there's no oxygen. So these organisms also require a low oxygen environment. So if you have an injury to a limb, let's say it gets crushed or punctured and it dies, the same thing with Clostridium tetani. So Clostridium tetani and Clostridium perfringens you can get from a puncture wound and then damaged tissue. So in this case, you have this necrotizing effect on your tissue, where necrotizing just means it dies and turns green and nasty and pus because of toxins produced by this particular organism. And then lastly, one that I mentioned previously, you want to keep out of your large intestine by keeping your Escherichia coli and your other fecal coliforms healthy, is Clostridium difficile. Make my eye a little bit better. So Clostridium difficile is responsible for pseudomembranous colitis. All right, I mentioned just colitis before, but it's a specific type of colitis. And this is the second time, at least I've mentioned C. difficile. There you have the rogues gallery, Bacillus anthracis, Clostridium tetani, Clostridium botulinum, Clostridium perfringens, and Clostridium difficile that are gram-positive rods that produce endospores. They're really, really difficult to kill. And particularly with the botulism and food poisoning, that's why you would need your pressure cooker.